Hi everybody, if you're watching this video, that means that I was unable to attend class. I'm going to run through the slides just as if I was there in front of you, or at least as close as that as I can do. So first we'll review last lecture. We talked about sexual reproduction, that each gamete provides one copy of each gene. We said that two gametes combine to make the diploid offspring, which means that now they have two copies of each gene. So when the gametes get made, you can pass along either copy of the gene. Okay, then we said that there were different versions of genes. They're called alleles. So for example, for hair color, you could have the recipe for brown hair pigment or the recipe for blonde hair pigment. Those would be alleles of the hair color pigment gene or the hair color pigment recipe. If an individual, if an individual has two alleles that are the same, then they're considered to be homozygous for that gene. If their two alleles are different, then they're heterozygous. Okay, and remember that's on a gene-by-gene basis. So, for your hair color, maybe you're homozygous. Maybe you got two copies of the recipe for black hair. But maybe for eye color, maybe you got one allele for brown eyes from one parent, and maybe you got an allele for blue eyes from the other parent. So that would mean you have the different alleles or different recipes for that one gene. Okay, and then to figure out what the phenotype, the actual physical appearance will be, that depends on what type of dominance it is. So we talked last time about classical dominance, meaning that one allele is the dominant allele and the other is the recessive allele. And the phenotype, the actual physical appearance, is going to resemble the dominant allele. The recessive allele is just not going to appear. It's almost as if it doesn't exist. It exists only in the genetics, but it doesn't exist in the actual, physical appearance. So let's say that brown hair is dominant over blonde hair. That means that if you got a recipe for brown hair from one parent, and you got a recipe for blonde hair from the other parent, your hair would actually be brown. So the blonde would not show up at all because brown is the dominant allele. So the genetic description is called the genotype. So that's the actual alleles that you have in your genetic code. The physical appearance, as we mentioned already, is the phenotype. Then we spent some time talking about Punnett squares, which were these, kind of neat ways to show all possible combinations of gametes that might occur. So you can get an idea of, the odds of each type of offspring. And we'll review some of those on the next slide. And then at the end we talked about pedigrees, which are those family trees that gave the genetic information. In a pedigree, the squares are males, the circles are females. Each pedigree is about a particular, genetic disease or condition. And so usually the shaded squares are the people that are affected with the disease. The unshaded squares are the people that are, considered, normal or unaffected with the disease. But on each pedigree, there's going to be labels that say, what each type of a coloring or a shading represents. Okay, so as always, if you do have questions about any of the stuff that we're covering, make sure you, ask me, shoot me an email if I'm unavailable, and if a different teacher, ends up filling in, then, make sure you ask that other teacher. Okay, so now we're going to run through a genetics example that's going to kind of summarize everything that we did last lecture. So let's say that we have a cross between these two parents, each of them with the genotype big R, little r. And then I've written down here that big R represents red, little r represents blue. So remember, with the abbreviations, you always take the first letter of the dominant allele, and then the capital version represents the dominant allele. The lowercase version represents the recessive. So even though blue starts with a B, it is still represented with the letter R. It's just the lowercase r, because that's the recessive version of this gene. Okay, so I want you guys to now, pause the video and try to write out the Punnett square that would represent this cross. Okay, and then try to figure out, what genotypes are possible, what phenotypes are possible from the cross that's listed right here, the big R, little r, crossed with big R, little r. So take some time, think about that. If you're still thinking about it, you should keep pausing the video, because now I'm going to start explaining how it works. Okay, so how Punnett squares work, which we talked about last time, is you're going to put the possible gametes from each parent on one of the sides here. Okay, so I have listed possible haploid gametes from parent 1 will be on the top, and the possible gametes from parent 2 will be on the bottom. And if you look at parent 1, doesn't matter which one you make as parent 1 or parent 2, but we'll just use the one on the left here. For this particular gene, it looks like they have one recipe for the red allele, and they have one recipe or gene for the blue allele, okay? Because gametes are haploid, that means they need one copy of each recipe, which means this person here can either make a sperm or an egg cell, a gamete, with big R or with little r. So we're going to list those as their possible gametes on the top here. Big R and little r. The other parent, in this example, it's easy because they have the same genotype, so they can also make gametes with either big R or little r. Remember, they don't get both because gametes are haploid. All right, and then you just fill in the square by writing, the appropriate letters that fill out each row. So we're going to take this big R and we'll write it here, and we'll write it here. We'll take this big R and we'll write it here, and we'll write it here, and then the same thing, little r's, write it here and here, and this little r would go here and here. So once we actually fill those out, this is what our square is going to look like. So hopefully you got that far, and then the question was, what type of genotypes and phenotypes are we going to get from this? So you have to look at each of these boxes, and this is the genotype written in the box, so that's easy. We know that we're going to get, out of 4, 1/4 of them, or 25%, is going to be big R, big R. 1/4, 25%, is going to be little r, and then half, 2 out of 4, is going to be big R, little r. If we were going to describe this with our other genetic vocab, we would say half of them are heterozygotes. We would say that 1/4 of them were homozygous dominant. Homozygous because they only have one type of recipe, dominant because it's the dominant recipe, the capital R. And then 1/4 of them are homozygous recessive, okay, because they have only one recipe, the little r, that's why they're homozygous, and they're homozygous recessive because little r is the recessive allele. Then we'd have to think about, okay, that's the genotypes, so that's easy, you just read it right off the square. You have to think about phenotypes. What would the actual coloring be of each of these? So if they're homozygous and they only have one recipe, that's the color. So big R is going to be red because big R corresponded to red. Little r is going to be blue because little r corresponded to blue. But then we have these two here, our heterozygotes. Remember, the heterozygotes have one copy of each of them. So that's where the question comes in. Are they going to be red or are they going to be blue? I didn't mention it, but hopefully you assumed that this is that classical dominance that we talked about in the last class. So that means that the heterozygotes are going to have the dominant phenotype. So they're also going to be red, even though they have a recipe, they have the allele here, the gene here, to make that blue coloring. Because this is classical dominance, they're just going to look red, the same as this guy. So our phenotype is going to be 3 out of 4 red, 1 out of 4 blue, which I have here. Genotypes, 1/4, 1/2, 1/4, and the phenotypes, 3/4 red, 1/4 blue. Okay, So today's lecture is going to finish up chapter 12. We're going to talk about some other types of dominance. So everything we've learned so far, classical dominance. Okay, these other ones are sort of specialty dominance that we're going to talk about. So when in doubt, assume, classical dominance, which is what we've already talked about. These are just sort of specialty scenarios that we're going to talk about today. Alright, so the first of those is incomplete dominance. So incomplete dominance is different from classical dominance, okay? If it's incomplete, it means that the heterozygote has a blended phenotype. For example, if there was a flower that had incomplete dominance, and let's say that the heterozygote had one copy of a red recipe and one copy of a white recipe, the heterozygote would actually look pink, so the phenotype would be pink. Remember that if we were talking about classical dominance, the heterozygote would either be red or it would be white, depending on what the dominant allele is. In incomplete dominance, it's always a combination of the two, okay? You blend them together. If we were using our previous example where we had the two colors of red versus blue, in that genetics example we did on the previous slide, and the dominant color was red and the other one was blue, if that was not classical dominance, if it was incomplete dominance, that would mean that the heterozygotes would show up as purple, which is a blend of red and blue. So incomplete dominance is sort of this other way that the heterozygotes can appear. Another thing to think about is polymorphism. This means that there's not just two alleles for a gene, that there's actually many different alleles for that gene. Okay, so this is an example here. You don't have to memorize, what's in the box. It's just an example that instead of there just being two alleles for rabbit coat color, it comes in this case in 4 different colors, and then you can see depending on what combination they have of these, they're going to end up with different phenotypes of their fur. So sometimes you can't just have, there's the dominant and there's the recessive phenotype. Sometimes there's several different types of phenotypes that can occur depending on how many alleles there are for the gene. So sometimes we talk about like foot size, and we say that you could have big feet or you could have small feet, and we pretend that those are the only two options. You either have a big foot or a small foot, but in reality, there's many things that are going to play into foot size, but let's say that there's, big foot and slightly less big foot and slightly less big foot and medium foot and slightly small and slightly smaller and smallest foot and teeniest foot, all these different alleles for foot size. It's not just, big foot or little foot. Of course, in reality, most of the things we talk about in this class, hair color, eye color, foot size, height, all of these things, they depend on multiple genes. It's not just a single gene, but anyways, keep in mind that there's not always just two choices of allele. Sometimes there's many, and that's called polymorphism. Another special type of dominance, so it's not classical, it's not incomplete, it's codominance. Codominance is a type of inheritance where you can have multiple alleles that occur all at the same time without getting in each other's way. And the best example is blood type, which is what we're going to talk about now. So for your blood type, there is a certain type of protein that appears on the surface of your blood cells. And there's 3 different alleles for this. Either you can have protein A on the surface of your blood cells. You can have protein B on the surface of your blood cells. Or you can have no protein on your blood cell, which is called the O allele. Okay, it's not an actual protein, it's just the absence of protein. So those are these 3 options, and Each person, of course, gets 2 copies of this gene because, remember, you get 2 copies of every gene because your cells are diploid. So you get 1 copy of this blood protein gene from your mother, you get 1 copy from your father. So you're actually going to have 2 copies. So let's see here. Whoops. So knowing that, I want you to take a minute and think. This isn't a multiple choice thing, it's just sort of a thinking thing. Think about how many possible blood protein genotypes there are. Okay, I'm going to reverse the slide. So we're trying to think of how many different genotypes there are. So if we have these 3 options of allele, A, B, and O, okay, and everybody gets 2 copies. So think of all of the combinations that can occur. Maybe pause the video here if you're gonna, think about it and try to write them all down. And then I'm gonna move on and explain it. So if there are 3 alleles, A, B, and O, and everyone gets 2 copies, we have to think about all of the 2-letter combinations there are here, okay? We can have 2 A's, we can have 2 B's, we can have 2 O's. Okay, if you got a copy of A from your mother and a copy of A from your father, this is what your blood cells would look like. If you got B from your mother and B from your father, this is what they'd look like. And if you got O from your mother and O from your father, this is what your blood cells would look like. Okay, and these are your blood types, type A, type B, and type O. Don't forget that we can also mix and match here. Okay, so what if you got type A from your mother, B from your father, type A from your mother, type O from your father, or type B from your mother, type O from your father, or vice versa here. Maybe you got the B from your father and the O from your mother. But either way, these are all of the possible combos here. If we have 3 different options and each cell has to have 2 of those options on it. And so the blood types for these other ones are going to be type AB, type A, and type B. So even though these last 2 here have this empty stick, the O, it's still considered type A. And even though this one is a B and an empty stick, it's still considered, whoops, sorry about that, it's still considered to be type B. The reason that this is codominance is because we can have the A and the B both appearing on the blood cell at the same time. We don't have some letter appearing here that's, half A and half B. We have the A protein and we have the B protein. So they can be codominant because they can both appear without impacting the other one. So they're not in each other's way. This one isn't, partially A and partially B. It's just A. And this one here, this protein on this stick here, is just B. So they're not in each other's way. So it's codominant, meaning that you can have both alleles in the phenotype. They don't have to blend. There doesn't have to be one over the other. They both win. Which is what it says right here. Okay, so this is less important about genetics. It's just sort of interesting, and I find that if I don't talk about it, people, ask about it. So blood type is one of those things that, you're familiar with everybody having a blood type. Some of you probably know what your blood type is. Some of you might not know what your blood type is. And you know that certain people can donate to certain people, and certain people can receive blood from people and not receive blood from other people. So we're just going to briefly talk about how you can receive different types of blood. So the whole thing with your immune system is that your body attacks things that seem foreign to you, because anytime you have an infection, that's like a foreign invader. You have a bacteria or a virus or an allergen or something that is inside your body that shouldn't be there. And if your body attacks things that you don't recognize, that means that anything that you're not familiar with gets attacked. Okay? And keep in mind here that whenever we see blood type O, these empty sticks, there's nothing there. It's just absence of something. Okay, so keep in mind that that's just sort of null. There's nothing there. So if your cells do not have any A protein on them, that means that your body recognizes that A protein as a foreign invader. So you cannot receive blood cells that have A protein. So if your type of blood does not have any A's on it, that means that you cannot receive A's because your body thinks that A is an invader. Same thing with B. If your blood cell does not have any B's on it, you cannot receive blood cells with B because your body thinks that those are invaders. So if we look here, if you are type A, okay, you have no B's, which means you cannot receive type B, you cannot receive type AB, you cannot receive the other type B, because they all have B's, and that would be something that's foreign to you. Your immune system would attack it. The opposite for type B, you couldn't get type A, you couldn't get type AB because it has the A, and you couldn't get this type A over here either. Because type O is just the absence of anything, it doesn't have A's, it doesn't have B's, that means anybody can get type O. So if you're type A, you could get, the double A, you could get the O, or you could get this one, AO, as long as you don't get any B's. If you're type B, you can get double B, you can get O, or you can get BO, as long as you don't get any A's. Now, if you're type O, that means that you are not familiar with A's, you're not familiar with B's, which means you cannot get double A, you cannot get double B, you cannot get AB, you cannot get AO or BO. If you are type O, you can only receive type O blood. If you are type AB, why don't you guys think about that for a minute? Think about what type of blood you could receive or not receive. Okay, if you are type AB, you're familiar with A because you have A's. You're familiar with B because you have B's, which means you can get any blood type. You can get just A's, you can get just B's, you can get O's, you can get AO, you can get BO. If you're AB, you're fine, you're cool with either A's or B's, so that means you can receive any blood. All right, so a little question for you guys then. And, obviously since I'm not there to, see your answers or anything, you're not even clicking anywhere, you're just watching this as a video, just kind of think of the answer on your own, and then, pause the video, try to come up with the answer, and then continue along. So pause the video, try to answer this. So the question, if you have the blood genotype AO, which genotypes can you receive blood from? Okay, so if you're AO, that means you're familiar with A, you're not familiar with B, and O, remember, is like nothing, so it doesn't matter. Which means that, oops, sorry about that. Come on, slide. You can receive AO, which is what you are, of course. You can receive AA, okay, because you're fine with A's. You cannot receive this one because it has a B in it. So our correct answer is going to be D, the one that has AA, OO, and AO. You could receive any of those types, as long as they don't have B's. That's the rule. Okay, so some of you have probably started thinking, well, wait a minute, what about, positives and negatives? Isn't that part of blood typing? It is. This is a different gene completely. So your blood type is determined by, these ABO proteins, and it's also determined by something called the RH factor. The RH factor comes in either the positive allele or the negative allele. Okay, the positive allele means that you have this factor on your cells. Negative allele means you don't have it. So you can think of it kind of like an empty stick. Okay, kind of like type O was sort of that empty stick. You can think of that for the minus allele too. If you have, and remember, for all of these, you're going to have two copies, 2 recipes, just like we said before that you have to have 2 of these proteins on your blood cells. So you can either be plus plus, you can be minus minus, or you can be a heterozygote, plus minus. These would be the 3 genotypes that are possible. And then down here at the bottom I have written, sorry this thing keeps popping up, that if you have a plus, if you have this plus factor, positive factor on your cells, then you're fine with it, you're familiar with it. If you do not have the positive factor, then your body would reject any blood that has that positive factor and reject it. Okay, so if you have the genotype positive, you can get somebody's blood that's negative because that's kind of null, it's nothing, or you could receive the positive. If you're negative, that means that you are not fine with this positive factor, so you cannot get the positive blood, you cannot get the positive negative blood. If you are positive, you're fine with the positive factor, so you could get either any type of blood here. When we describe these, we describe them as either positive if you have the positive factor, so that would be either this one plus plus, or this one plus minus. Even though they're the heterozygote, we still just call them positive. That just means that they have the positive factor on their cells, they're familiar with it, they can accept positive factor. So keep that in mind. It's sort of the secondary part of blood typing. Alright, so let's switch gears here. There's these 2 important vocab words for genetics. They're wild type versus mutant. Genes come in multiple alleles, usually, and all of these are because of random mutations that have occurred over time. Mutations are happening all the time, just, they're small things. It's not like it's not X-Men, someone doesn't get a mutation and that causes them to suddenly grow a giant pair of wings. That would require lots and lots of mutations over time, because wings themselves, there's a lot to them. There's bones, and there's hollow bones, and there's feathers, and blood vessels, and muscles, and all of these things would somehow have to appear. So it would take many mutations in order for somebody to suddenly just sprout wings one day. But there are little mutations that happen here or there. Excuse me. So because genetics are changing all the time, scientists tend to call the most common allele the wild type allele, and then anything else is a mutant allele. Okay, now we usually don't use the word mutant when we're talking about genotypes in humans, because it sounds like it's negative, but it doesn't. When we talk about it in science, it's not something negative. It just means that you have the phenotype that is, not the most common one in the rest of the population. So the wild type allele is just the one that's most common. It doesn't mean that it is causing something or to be wild. It doesn't mean it's ferocious or it's uncivilized. It's not wild. It just means it's the allele that is out there and is the most common one, and whatever sort of the new version is, the specialty version, that's the mutant allele. Okay, something else to think about. So not every gene is going to be expressed in every cell. Remember, every single cell is going to have the entire genome. Okay, so when we replicate our DNA, we replicate the whole thing, and every cell gets it. So that means that if we were to take a skin cell, and we were to take a brain cell, and if we took the DNA out of the skin cell and we took the DNA out of the brain cell, they each have the full recipe book. It's not like the brain cell just has recipes for brain, or the skin cell just has recipes for skin. Each cell, every cell in the body, has every single recipe. The difference is which recipes they use. In the brain, they're not going to be expressing or making or cooking those recipes to make skin cell things. They're going to be using the recipes for brain cell type stuff. the opposite for the skin cells. So sometimes there can be, trouble in genetic paradise if the genes are expressed where they shouldn't be, or if they're expressed at different amounts than they should be. So remember, all these genes are just recipes. And there's no harm in having a recipe if you don't make that recipe. But if you start making a recipe where you shouldn't be making it, or at a level you shouldn't be making it, then there's trouble. So this picture here is kind of neat. On the left we have a fruit fly, and these little sort of wispy things that we can see coming off his head, those are regular antennas. Okay, but this guy on the right here has a problem where in his antenna cells there are things being expressed to make his legs. Okay, so something happened and there was a misdirection, and they started making the recipes for legs instead of making the recipes for antennas. So they expressed the leg or the foot genes up here. So the reason these are kind of thick and different looking is because these are, little funny legs that stick out of its head. Another thing to think about is that sometimes the dominant allele is the bad guy, is the bad one. So there's a disease that, we're talking about on this slide here. That's what the picture's from. It's called Huntington's disease. And it is caused by the dominant allele, which means that if you have one bad allele, you're going to end up having this disease. So sometimes we think of dominant alleles as being the better ones or the more common ones, and we think recessive ones are maybe weaker or rarer, but that's not always the case. So Huntington's disease, obviously the majority of people in the world do not have Huntington's disease, which means that all of us that don't have it, I'm going to assume that I don't have it, although it's something that comes on late in life. So, sometimes people don't know they have it, until later when they start having symptoms of the disease. Anyways, if I assume that I don't have it, that means that I, and everyone else that doesn't have it, is going to be homozygous recessive. Okay, so it doesn't always mean that things that are recessive are rare. It doesn't mean that things that are recessive are, the weaker version or something like that. This is just an example where the dominant allele actually causes the disease. Of course, there are some diseases that are caused by recessive traits. This is an example here of a kid that has albinism, which is a recessive trait, okay, which means that anyone that does not have albinism is either homozygous dominant or a heterozygote. And this kid here that has albinism, which is a skin pigment abnormality, is going to be homozygous recessive and have albinism. So different diseases can be caused by different things. Some can be caused by dominant alleles, some can be caused by recessive alleles. All right, question for you guys. So again, you should pause the slide, try to answer this, and then I'll explain it. So it says, if a disease caused by a recessive allele, which people will have the disease phenotype? And if it's a recessive allele, and if we assume classical dominance here, that means that the people that are going to have that phenotype are going to be homozygous recessive, okay? Because if you are homozygous dominant, you wouldn't have the recessive allele at all. If you're heterozygous, that means you got one copy of the dominant allele, you got one copy of the recessive allele, but it means that the actual phenotype would still be the dominant phenotype. So the only one that has that recessive phenotype is homozygous recessive, 2 copies of that recessive allele. All All right, so we've said many times that humans have 23 chromosomes, and then they have 2 copies of each, so they have 23 unique pairs of chromosomes. We haven't talked much about the difference between chromosomes 1 through 22 and then chromosome 23. So 1 through 22 are called autosomes, and they each have one length, meaning chromosome 1 is one length, chromosome 2 is one length, etc., etc. The 23rd chromosome is called the sex chromosome. Most of the things that are, I take that back, not most of the things, but quite a few of the things that are on this 23rd chromosome have to do with reproduction and sexual development type things. And this chromosome is special because it comes in 2 different varieties, okay? The X, which is this longer variety, or the Y, which is this tiny, tiny little guy right here. Okay, so you can either have 2 copies of X, or you can have 1 copy of X and 1 copy of Y. The sex chromosome is the 23rd pair, and it comes in X or Y. Biological females have 2 X's. Biological males have 1 X and 1 Y. The Y chromosome doesn't have much on it except for male reproductive stuff. The X chromosome has some stuff that, both males and females need, and it has, a lot of the female reproductive stuff on it. If things are mutated on these chromosomes, that often can cause problems with formation of reproductive organs, hormone production, infertility problems, things like that. All right, question for you guys. So again, pause the slide. Since each gamete gets 1 copy of the sex chromosome, what are the odds that a couple will have a boy? Okay, so think about what the genotypes would be. And then think about what are the odds that a couple has a boy. So if you did a square, your square should have XX on one side, because females could pass along either an X or an X, because females are XX. Okay, so one side of your square would have XX. The other side of your square would have XY. And then if you fill those out, you'll find that half of the little squares end up being XX and half end up being XY. So the odds that a couple will have a boy is 50%. And interestingly enough, the sex of the child is determined by the sperm, because the female egg always contains an X. That's the only type of chromosome they can bring to the party is an X, because they are XX. The males are the ones that can either supply the X or they can supply the Y. So about half of sperm are going to have an X in them, and if one of those sperm is the winner that fertilizes the egg, it's going to become a girl. And if the other half of the sperm are going to be containing a Y, and if those sperm are the ones that win and fertilize the egg, then the child will become a boy, at least biologically female, biologically male. It used to be a long time ago that women tended to be penalized or ostracized if they were not producing enough male offspring. But the whole time it's only up to the father and the father's sperm. So guys, whichever of your sperm end up being the winner that fertilizes an egg someday, assuming that you have children, maybe you won't. But yep, that's going to determine if it's a male or a female. Okay, so next topic here. So we talked about these sex chromosomes, and because females get 2 X's and males get 1 X, that means that males don't get 2 copies of the things that are on the X chromosome. And the X chromosome has some interesting stuff on it. It's not just reproductive genes. The X chromosome is a large chromosome, so it has stuff that males and females need. But obviously, the males are only getting one dose. So they're not diploid. They have one dose of everything that's on the X chromosome, or they only have one recipe for everything on the X chromosome, whereas females, because they have two X's, they're diploid, okay, and so they would end up with two recipes for everything on the X chromosome. The picture here is showing fruit flies, and in fruit flies, eye color is on the X chromosome, and we can see that there's all these different eye colors. For those of you not familiar with fruit flies, these things that are red looking, that's the eye. So in this guy here, you can see the eye is kind of white. This one is this real dark color. This one's pretty dark here, but these are their heads that have these big eyeballs stuck on them. And it's different. You can get all these different eye colors. If we look at this square here, let's say that big W are the dominant red eyes, little w are the recessive white eyes. Females, remember, because females are diploid, they could either be big W, big W, big W, little w, or little w, little w. So the same as everything we've talked about, with just classical dominance for females. But males, they only get one dose of W because their other sex chromosome is the Y, and the Y does not have this gene. Okay, so we could even kind of ignore these Y's here and just think that the genotype for male is either big W or little w. Since males only have one copy, they're often called hemizygous. So they're not homozygous or heterozygous because they only have one copy. Hemi means half, so they're half zygous. They only have one copy, they don't have two. Anyways, if we look at our chart here, the arrow pointing up and outward, that's the male symbol, and the hanging down cross, that's the female symbol. And so this male here, he has X and he has Y, and on his X he has big W, that's the allele he has. The female, she has two X's, and she's a heterozygote, okay, because she's got one little X, she's got one little W and one big W. I'm sorry, this is kind of blurry, but we can see it down here in the larger version. So here's the possible male gametes. The male can provide the Y, or the male can provide the X that has the big W. The female can provide either of those X's, X with little w or X with big W. So then if we fill out the chart here, and I guess in this chart it still isn't that easy to see, but down here we can see the little w's versus the big w's. Maybe I think the female, I take back what I said, I wish this was clearer. Maybe the female, the female has white eyes, so each of these W's must be a little w. And so each of these X's here has little w. That was my mistake. I thought that one of those was a big W, but okay, so the female can only provide X's, and those X's only have the little w. The male has the big W or the Y. All right, so if we fill this out, this one got an X, and it got a big W, and it got an X, and it got a little w. Because it has two X's, it's female, the cross hanging down, female. And because it got that dominant big W allele, the eyes are red, because big W is dominant red eyes. In this box over here, we have, again, we got that X with the big W from dad, and we're XX, which means it's a female. And then it got that X with the little w from mom, so it's also a heterozygote. So both of these top ones are females, and they're heterozygotes. And they have the phenotype for the dominant red eyes. The bottom row here, they got the Y from dad, so both of them are going to be XY, so that's why they have the male symbol here. They're going to be male. And because the X that they got from mom, if they got this X, this guy here, is the little w, he has white eyes. And this one got this little w, which means he has white eyes, because that's the only one he has. Even though that's the recessive allele, because males are hemizygous and they only have one copy of X, that means whatever they have on that X, it doesn't matter if it's dominant or if it's recessive, that's the phenotype that they get. So these are all the possible options. All right, so here's a question. So what's the likelihood that a boy will have a recessive X-linked disease if the mother is heterozygous and the father is homozygous dominant? So again, pause the video and try to think about it. If you need a little more information here, a recessive X-linked disease means the disease is on the X chromosome, and those people that have that recessive genotype would have the disease. Okay, so in the picture here, we can see that we've put the mother on the top, and she is heterozygous. So I've got two X's there. One of the X's I have marked as having the disease, and the other X I have marked as not having the disease. And then the father I have listed as homozygous dominant. As I'm reading this, I'm realizing that it should say that the father is hemizygous dominant, okay, not homozygous, hemizygous dominant. And in the picture here, we can see that he has his X and his Y, and his X has the dominant allele. Okay, so now we have to think about if the mother can contribute either a diseased X or a non-diseased X, and if the father can contribute either the Y or the X that has the disease on it, what's the likelihood that a boy is going to have the disease? So first of all, for it to be a boy, dad has to donate his Y. Okay, if dad donates the Y, then we just have to wonder, what is the mother giving? So she is heterozygous, so she can either give the diseased X or the non-diseased X. So it's going to be 50-50. If we look at just the boys, half of them are going to get the good X from mom, and half of them are going to get the diseased X. So this is going to be C, 50%. The father is hemizygous dominant, so he had the Y and he had the dominant allele, which was the unaffected. The mother, she had two X's and she was a heterozygote. So this is the same example from the previous slide. So if the father is normal, that means that the son, if they have one, is going to be 50-50 having the disease or not having the disease. A daughter, which we did not talk about on the previous slide, a daughter will never be affected herself, but she'll have a 50% chance of being something called a carrier. A carrier is someone who is possible of passing on the disease but doesn't actually have the disease themselves. So if a mother is a heterozygote and she herself does not have the disease, but if she does have one copy of that disease allele, she's called a carrier because she's not sick herself, but in her genes she has the capability of passing on that diseased gene to one of her children. So these were the 4 choices here. We said of the boys, if the boy gets the good X from mom, he's unaffected. If the boy gets the bad X from mom, then he's affected with the disease. And the girls are always going to get dad's X, and if dad is unaffected, that means they always get a good X from dad. They don't get the Y from dad, because if they did, they would be he's and not she's. So they're getting the good X from dad, and then they're getting either mom's good X, that would be this daughter, completely unaffected and has 2 great X's, or will be a carrier. Unaffected because she's a heterozygote, so this good X means she doesn't have the disease because it's a recessive disease, but a carrier because she still has that one chromosome that she could pass along to her children. Okay, so one thing to keep in mind, so there's 22,000 genes in the human genome. We get 22,000 genes, recipes, from our father, and then we get 22,000 from our mother, okay? Every gamete contains 1 copy of every gene, which means inside your little egg cell or your sperm cell, you are providing 22,000 recipes. It's crazy to think about. But because 22,000 is too many to think about, we've so far, in this, we've only been pretending that there's, 1. Okay, so far we've just done 1, and we've, mentioned, maybe there's another one out there. But anyways, now we're going to do an example where we're looking at 2 genes at the same time. Okay, so remember, your egg or your sperm is going to have 22,000 recipes in it. We're going to talk about there just being 2 and how 2 different recipes can be passed along, 2 different genes. So we're going to go back to peas, back to Mendel, and one gene we're going to think about is pea color. The other gene we're going to think about is pea texture. Pea color can come in yellow or green. Yellow is dominant. Pea texture can come in round or wrinkled. Round is dominant. So remember, each gamete gets 1 copy of each gene, okay, which would mean that each sperm or each egg, each gamete is going to have 1 color recipe and 1 texture recipe. Okay, so since we're looking at 2 genes now, the genotype for an individual, for a person or a pea plant, I guess in this case, is going to be just in this example is heterozygote for color, so they're big Y, little y, and they're homozygous dominant for texture, big R, big R. Okay, so that's their genotype if we're considering just 2 genes, which means that they can pass along for color either the big Y or the little y, and they can pass along for texture either the big R or the big R. So if we pair together all of these in different pairs, let's start with big Y with big R. They could also pass along the big Y with the other big R. They could pass along the little y with the first R or the little y with the second R. So far, we have only done single genes, which means that each gamete we make always has just 1 letter in it. Okay, but because these are 2 different genes, we need a recipe for each. A pea has to have a color and a texture. So we need to have a recipe for color and texture inside each gamete. Okay, so if you're looking at 2 different genes, make sure that you put 1 copy of each of them into the gametes that you're making. Because some of these are redundant, the first 2 are both big Y with big R, and the second 2 here are both little y with little r. There's 2 unique gametes that are possible. So of this genotype, we could either have big Y, big R, or little y, big R. So those are the two gametes that this individual can make. If we change it, okay, so notice the slide changed, but not much changed on it. So now we change the genotype up here to big Y, and big R, little r. So now think about all the different combos we can make. Okay, we could pair the big Y with the big R. We could pair the big Y with the little r, which is what we see here. We could pair the little y with the big R, or the little y with the little r. Okay, so now it's 4 possible unique gametes that can be made from this parent. So whenever you're looking at 2 genes, always make sure that each sperm or each egg, each of your gametes, gets 1 copy of each gene. Alright, so from this genotype, big D, big D, big B, how many unique gametes can be made? Well, remember that each of them has to get 1 copy of whatever the D gene is and 1 copy of the B gene. So for the D gene, they can contribute big D. From the B gene, they can contribute big B. So big D, big B is the only possible gamete that can be made. So the answer to this one is 1. Okay, now think about what offspring combinations are possible if you cross 2 parents that are heterozygous for both genes. Okay, so you can, well, I was going to say ignore the top, but I'll walk through it real quick. The top here shows a parent that is homozygous dominant for both genes being crossed with a parent that's homozygous recessive for both genes. The outcome are these offspring here, the F1s, which are heterozygous for the color gene, big Y, little y, and heterozygous for the texture gene, big R, little r. So the question up on top here in green is saying, if we cross the F1s with each other, okay, heterozygote at one gene and heterozygote at the other gene, if we cross these with each other, you know, then what outcomes do we get? So the Punnett square here is if we take the possible gametes that this can make, big Y, big R, little y, big R, big Y, little r, and little y, little r, and because we're crossing it with the same thing, we have the same gametes written over here. Okay, so if we cross these F1s with each other, these are all the possible outcomes that we get. You fill out the square the same way, even though your gametes have 2 letters. You still just fill out the letters. So this was big Y, big R. This was big Y, big R. So you just write that in the square where they both meet. Okay, like this one over here. We took big Y, we took big Y, so that's why it's big Y, and then this one had little r, and this one had little r. So the box where they intersect at is big Y, little r, little r. Okay, and then we have to look and figure out what the phenotypes are. So if we know that anything that is big Y, okay, if we know that anything that is big Y, or anything that is big Y, little y, that's going to be yellow, because yellow is dominant. The only ones that are going to be green are the ones that are little y, little y. And you can see if you look at all these green ones, they're little y, little y. So that's for color. And then we have texture. So the texture was that round is going to be dominant. So anything that has big R, homozygous dominant, or has big R, little r, a heterozygote, those are all going to be round. And then the ones that are going to be wrinkled are the ones that have little r, little r. So you can see the ones that have this wrinkly shape are little r, little r. Okay, then we have this one on the bottom here, little y, little r, little r. We can see that that one is both green and it's wrinkled. And if we actually count up the different categories here, we see that 9 of them are going to be yellow and they're going to be round. 3 of them are green and round. 3 of them are yellow and wrinkled, and only 1 one is going to be green and wrinkled. So this is called a dihybrid cross. It's written up here, dihybrid cross. Di because we're looking at two genes, and hybrid because they're heterozygotes for each of them. So they're a hybrid for the Y gene, they're a heterozygote for the Y gene, they're also a hybrid for this R gene, and di because we're looking at two genes together, and they're a heterozygote at both positions. So that's why it's called a dihybrid cross. Okay, I know this is getting to be kind of a long lecture, but because you're watching it on a video, hopefully you've had a chance to, pause it here and there and take a little break. My voice is giving out a little bit, but I'm just going to keep going because we're almost done. So, linked genes is something else that we need to talk about for genetics. So, linked genes are genes that are both on the same chromosome. So if you think about meiosis and think about what things get separated into gametes, we're separating chromosomes into gametes. We're not separating genes into gametes because there's 22,000 genes. It would be too difficult to separate them all individually. So instead, what we do is we just make sure that each gamete gets one copy of each chromosome. And then we, kind of assume, okay, that means they get one copy of each gene because there's only certain genes on certain chromosomes. But things that are both on the same chromosome are always obligated to kind of travel together during meiosis. Okay, so they're called linked genes if they're on the same chromosome. So we're going to go through a little example here. Using rabbits, and let's say that they have a gene for ear length and fur color, okay, and we're going to assume unlinked, and then we'll talk about if they're linked. But if we assume unlinked, if a rabbit was making gametes and these were the diploid cells, think about what gametes this rabbit could make. So remember, a gamete is haploid, and they get one copy of each chromosome. So of this long chromosome here, they've got red or blue version, and the gamete can get either of those. Okay, the same thing for the short chromosome down here. They can either pass the gray one or they can pass the white one. So if they package these together in gametes, you could either have long ears paired with white color, you could have long ears paired with gray color, you could have short ears paired with white or short ears paired with gray. So those are the possible gametes that can be made. Now let's pretend that ear length and fur color are linked. Okay, so now I've put the long ear with the white color on the same chromosome. I've linked them. And the same thing with the short ear and the gray color. I've linked them. So now if we think about what gametes can be made here, now we only have one chromosome. So that means that the gametes can either be long ears with white or short ears with gray coloring. So there's only two options. The long and the white are obligated to always go together because they're on the same chromosome, and the short and the gray always go together. Maybe some of you have already, kind of jumped ahead and thought, well, wait a minute, what about recombination? So that's exactly what can happen to kind of swap the long and the gray and the short and the white. Okay, so remember we had them on the same, and we said that either long and white travels together or short and gray travels together, but recombination is that process where we can actually swap those homologous chromosomes. Okay, so that's the only way that we can unlink the alleles. So that means that if we have crossing over, we could actually cross over and swap so that long and gray are on one chromosome and short and white are on one chromosome. There's something called recombination frequency, and this is important because it can tell us how far apart genes are located. So when you think about recombination, we just kind of say, oh, they swap bits of DNA. But physically, they have to actually, kind of wiggle their way over. They have to attach. There has to be sort of cutting and pasting involved so that you can actually swap bits of your DNA. If genes are located far apart, like this example on the left, all these X's here represent physical space where there is room for the top chromosome to reattach to the bottom and the bottom to reattach to the top. The example on the right here shows that if the genes are close together, here we have A and B closer together, there's just this tiny little space here where you can actually fit in the cutting and pasting and machinery that you have to do to have that crossover event. Okay, so in order to get this part with big A attached to this part with little b, you have to fit one of these crossovers and all the stuff that goes with it in between A and B. Okay, over here there's only this tiny bit of space where we can make big A attached to little b, instead of being attached to big B. And if they were even closer together, there wouldn't be room. There wouldn't be space where we could go from big A and reattach it over here so we can attach to little b. So these crossover events where recombination occurs, there's physical space that's required in order to do that. And if things are close together, you can't, have these recombinations between those genes. So if genes are far apart, they are more likely to have recombination, between them, to have the swapping. If they're close together, it's not likely that you're going to be able to swap the alleles. Oh, I guess my question got cut off at the top. So I'll just kind of read it. So if these are the recombination frequencies, if A and C recombine 18% of the time, if B and C recombine 25% of the time, and if A and B recombine 6% of the time, what would the order of these genes be? So if we know that these are the recombination frequencies, which of these arrangements on a chromosome is going to be correct? Okay, based on those frequencies. So you can pause the video and think about it. So remember, recombination frequency is going to increase if the genes are far apart, and if the genes are very, very close together, it's going to decrease. So we can first look at which two should be the furthest apart. So B and C had recombinations between them 25% of the time. That's the biggest number that we see here. Our choices here are 18, 25, and 6. So 25 is the biggest we see. So we want to make sure B and C are as far apart as they can be. So that means we're going to narrow it down to either choice B or choice D, because that puts B and C far apart. Now we have to look at these other things. Let's look at which one should be closest together. A and B should be the closest together. So now let's look. We said that we were down to between B and D, and A and B should be closest together, closer together than A and C. So in choice B here, here's B and here's A, okay, close together. A and C further apart. And if we look at choice D here, that one has A and C being close and A and B being far apart. Okay, so choice B is going to be correct because it has the shortest distance between A and B, the next shortest distance between A and C, and then the largest distance between B and C. So choice B is going to be correct here. Okay, another important genetic vocab word, I know you're probably sick of them, is epistasis. So epistasis is when the phenotype of one gene affects the phenotype of another. In this example here, they have this mouse, coloring, and you can either have this like mottled, which means kind of like a patchy coloring, or you can have solid coloring. And then a completely different gene has the ability for pigment production. So when we look at these mice here in the picture, we can see that some of them are going to have mottled fur, kind of patchy fur, and they're the ones that kind of look brown. And some are going to have more of this gray color, and then some of them have the white color. So the white color is because they don't have any pigment produced, so they have the mouse version of albinism. So these are albino mice. And some of them are supposed to have that patchy coloring, that mottled agouti coloring here, but they show up as white. They don't show up as this sort of patchy brown color because they're albino mice. So they don't produce any pigment, so it doesn't matter if the pigment they were supposed to produce was supposed to be this mottled, or if they were like a solid black or gray color. I find that the mouse example, it's nice because you can see the genotypes here, but what I have written at the bottom here will probably make more sense to most of you. So if epistasis means that the phenotype of one gene can kind of interfere with the phenotype of another, let's think about hair texture and baldness. So let's say that there's straight hair and there's curly hair, and it doesn't matter if you have the allele for straight hair or if you have the allele for curly hair, because if there is a different gene that says hair production, yes or no, and if you have the baldness version of that other gene, it doesn't matter if you have the allele for curly hair. If you are bald, you are not making curly hair because you're not making hair at all. So that's epistasis, where you might have a you might have a genotype, but that phenotype doesn't show up because you have a different phenotype that is getting in the way of that. So that's what epistasis is. Okay, I believe this is the last slide that I have here. Mendel's laws. So these are things that we've talked about, but we haven't actually given them names. I find that in some genetics courses you start by saying these are the laws and these are the names, but I think that after we explain a lot of stuff, it's easier to understand the laws, so I always just put them at the end. Mendel's laws, one was the law of segregation. This means that if you have 2 copies of each gene, which we do, when you make gametes, those 2 copies segregate or separate into different gametes. Okay? In other words, gametes are haploid. So that means that if you have 2 copies of a gene, which you should have, when you make a gamete, you're not going to get both of them in one gamete unless there's a mistake. If you have for hair color either the red hair color or the black hair color, you're going to make a gamete that has either red hair color or black hair color. So your red hair and your black hair recipes are going to go their separate ways when you make gametes. That's the law of segregation. It's just the idea that gametes are always haploid. They only get 1 copy of a gene. Mendel's other law, the law of independent assortment, is that the pairs of homologous chromosomes split up independently in meiosis 1. So in class I had talked about this, where I had said, maybe the blue one goes to the left, maybe the red one goes to the right, and that for each pair they decide that. It's like they get together and flip a coin and say, do you want to go left, do you want to go right? So for each pair of homologous chromosomes, they split up independently. So for chromosome 1, the long chromosome, maybe the mother version goes left and the father version goes right. Maybe for chromosome 2, they do it the same, the same way. Or maybe the father version goes left and the mother version goes right. Maybe they do it the opposite way. And all of those pairs are going to split up independently. So that's what the law of independent assortment means. Unfortunately, the way that Mendel named his laws, I feel like if he named the first law, the gametes are always haploid law, everyone would understand it. But instead, it's called the law of segregation. And the second one here, if he would have said the law of homologous chromosomes splitting up independently, people would understand that. But instead, it's called the law of independent assortment. So make sure you know Mendel's laws, the names of them, and then what they actually mean. Okay, this is my last slide. So if you have any questions, again, please contact me. I know it's maybe difficult to just watch the video on this instead. The benefit, of course, is you can pause it and rewatch it, whereas if, in lecture, in class, maybe you can't have me explain something twice. But I hope that you're understanding genetics, and I hope that I see you soon.